All right, well, thanks everybody for coming, especially for those of you that are coming in person. The one most important thing I'm supposed to mention is that the CME number, the secret number, is 57657. And there'll be a slide about that, I think, also. So my name is John Satterley. I'm a program officer at NIDA, and I'm also a member of the Common Fund for Denucleome program. And so on behalf of Ananda Roy from the Common Fund program and the Common Fund for Denucleome program, we're really thrilled to have Jen as a WAL speaker today. So Jennifer Phillips-Cremins is an associate professor in genetics and biochemistry at the University of Pennsylvania. And Jen has had a very exciting career trajectory, even if you just look at the grants that she has. So for example, she obtained an NIH New Innovator Award, then a Common Fund for Denucleome Phase 1 Award. More recently, she's received 2 additional Common Fund for Denucleome Awards and the prestigious NIH Pioneer Award, among other grants. So what I would like to say about that is that Jen is a really good example of how NIH and the Common Fund can serve as an incubator space, both for great science and for great scientific talent. So on with the exciting science. Today, Jen is going to tell us about the science of connections, bridging chromatin folding, synaptic plasticity, and neurophysiology. So take it away, Jen.
Hello, everyone. It's a great privilege to be here. I want to thank John Satterley as well as my colleague Ananda Roy for the support over the years and for inviting me here today to give this lecture. I'm excited to be here because I'm going to talk to you about something maybe that you didn't expect for me to raise, and that is the question of memory. Memory is important to all of us. It underlies our ability to read, to write, to acquire new skills, to maintain relationships. It's an important question because it's a unifier of the human condition. No matter our background, we all hold space to carry with us wonderful memories. And we also carry space with us to remember very difficult events, for example, isolation, loss of a loved one, or trauma. Loss of memory underlies diseases such as Alzheimer's, and exacerbation or resilience of fear memory is critical for disorders such as PTSD. So what encodes memory? Sort of one leading line of thought is that it lies in neurons and the connections between them, their synapses. In the mammalian brain, there are billions of neurons connected by trillions of synapses. And what's so interesting about these connections is that they're not static, but rather dynamically altered in a continuous manner across the lifetime of an individual in response to experiences. So I'd like to show this video of data. This is from Jim McGee's lab because it highlights the difficulty and excitement of the problem space for scientists that are studying memory. Here, Jim and his colleagues are using a calcium sensor to track longitudinally action potential firing in neurons in the mouse brain. The mouse is housed in the dark and then exposed to a sensory stimulus. And this sort of highlights just how difficult it's going to be to understand what underlies and maintains memory over the long term, because clearly these neurons are changing over time, and even the same neuron might be having different patterns of stimulation temporally. So we like to take what I call a multi-scale approach, and the idea is that the brain is regulated at many different biological scales. And part of the complication in finding patterns that lead to biological insight through all of these challenges is that these scales also must be integrated to understand how neural circuits work. Of course, there's large-scale anatomy, there's physical connections between neurons, and also labeling of so-called engrams that have had previous firing of action potentials. There's various neurophysiological patterns, both structural and biochemical, synaptic plasticity. And then, of course, in the nucleus, where we think a lot about the post-translational modification of chromatin, higher order folding of the chromatin fiber, and even variation in the genetic sequence, which among individuals might contribute to differences in circuits. And so across these scales, we aim to sort of study maybe 2 or 3 model systems and begin to gather this information in those model systems and integrate to lead to insight. And this is where we're going forward. And I have to say that over the first 10 years of the lab, where we've really focused is on the nucleus, and now we're heading out to the synapse. And so where we focused is on understanding so-called epigenetic or chromatin modifications to the genome, in which there are hundreds to thousands of various chemical modifications, either directly to the DNA or onto histones, that are modified through experience and also dynamically altered throughout the lifetime of a circuit. And so the classic approach to say what aspect of so-called chromatin marks is functional is to do a type of genome editing experiment and then assess how does that particular loss of function mutation affect expression of a nearby gene. And over the years, we've taken a bit of a different, what I would call structural approach, in which we have mapped the higher order folding of the chromatin fiber within the 3D nucleus. We've computationally identified the spatial or physical touchdown points between distal DNA fragments. And then we've studied how genes are regulated from long-range mechanisms. So when I started in this field as a postdoc, all the way back in 2008 or so, this was the technology and where the field was at that time. We could use a version of DNA fish to paint chromosome territories and confirm that they were in independent locations in the nucleus. And then at the other end of the spectrum, we could also use DNA fish and pairwise probes to look at focal dots in the nucleus and to begin to make inference about the overlap of the dots and that it would represent sort of a long-range looping interaction between two distal fragments. And everything else in between was sort of theoretical or abstraction of possible folding patterns. And so what I think I'd like to say here is that over the last 10 years or so, it's just been a wild ride, I think, from all colleagues in the 4D nucleome and then also internationally, where now with the advent of many different forms of technology, we've been able as scientists to make new discoveries to fill in the gaps, confirming territories, but also finding A-B compartments, subcompartments, even nanocompartments, topologically associating domains, subtads nested within tads, even nanotads, and also long-range looping interactions. So for those that are in a sort of broader audience today, I wanted to show you an example of the type of data that's been able to drive discoveries in this field over the last 10 years. This is a so-called chromosome conformation capture map generated through principles of molecular proximity ligation, originally published by Joop Dekker and Nancy Kleckler's lab back in around 2002. And the idea is that if you post the genome fragment of interest on both the X and the Y axis, one can look at the pixel intersecting those particular genomic loci and look at the reads in that pixel. That represents the number of times that those two particular fragments were detected as artificially glued together after this molecular assay. And this can be a readout of the frequency of the two fragments' contact with each other in physical or nuclear space. And so what one can see immediately is that there's very exciting and also very difficult to understand folding patterns that these maps revealed. And one I want to highlight are these so-called focal dots. These dots have been very difficult and taken almost 10 years of work, both experimentally and computationally, both to detect and to analyze. But now as a community, we have the ability to detect them genome-wide. And what they represent biologically is a so-called long-range looping interaction in which two distal fragments physically contact each other at high frequency in a population of cells, oftentimes mediated by architectural proteins, such as shown here, CTCF, and created through the mechanism of loop extrusion, where cohesin in an ATP-dependent manner extrudes out the intervening DNA until it abuts up against these CTCF-mediated boundaries. So when you see focal dots of this nature, they're important because they represent a so-called long-range looping interaction. So when my lab started, we could see that the technology was advancing rapidly, and we knew that reference maps were beginning to be created across a wide range of cell types. And so the question that we had is, what if we sort of moved to the next thing that was going to be difficult? And that is, if we know that the genome folds into many complicated structures, How do we figure out what aspects of structure are truly functional and give us insight into what the genome is doing? And I wanted to share, this actually came out today in the meeting with the students and postdocs over lunch. How does one pick a biological problem? And I have to say for us, what we tried to do is to find a question that is timeless. And so I'd like to use this example of the earliest possible reference that I could find. This is from Thomas Hunt Morgan, back in what I would call the good old days where nature papers were single page and only one hand-drawn figure. There was no social media, but there was clearly no absence of snark at that time, because Thomas Hunt Morgan writes that Castle has proposed an arrangement of linked genes in 3 dimensions based on the assumption that the distance between any 2 loci is exactly proportional to the observed crossover value. He believes that this system gives a better agreement between observation and expectation than does the hypothesis of a strictly linear arrangement that we have developed. And so I think probably John can speak to this, as well as many in the 3D genome field. I see Pedro here, that these types of debates are still ongoing right at this exact time, advancing all the way to 2024, and will certainly be happening at the closing meeting of the 40 nucleome phase 2. This question truly is timeless, and there is just so much more to discover for the next wave of scientists coming after us. And so over the years, we've conducted a variety of studies, first in models of neurons through neural differentiation, neural reprogramming, and more recently, neural circuit stimulation, both in vitro and in vivo. And what we found is that indeed loops are highly dynamic across all of these systems, most importantly, changing on very fast timescales during activation of neural circuits, and also necessary and essential for the activation of critical synaptic, immediate early, and secondary response genes, both in vitro and in vivo. Over the years, we've also developed experimental and computational tools, and here synthetic architectural proteins to get at the genome structure-function relationship. And this is an example where we've created something called light-activated dynamic looping to form loops on demand and ask if they were sufficient for enhancer-promoter activation of gene expression. And then in wondering if perhaps loops might not matter for gene expression, or maybe they mattered for replication origin placement, we worked in collaboration with the Gilbert Lab to understand that cohesin-mediated loop extrusion was both necessary and sufficient for the placement of replication origin machinery at specific locations in the genome in early S phase. I just wanted to highlight, these are all now alumni who worked in these early stages. John Began and Thomas Gilganas, both now having high-flying careers in startup companies, both in artificial intelligence as well as in the development of therapeutics. Dr. Ji-Hyun Kim and Mayuri Reggie, who are both my first postdocs in the lab, and now they each run laboratories in South Korea as well as in Mumbai. And then more recently in the replication study, Dr. Dan Emerson, who's a postdoc at CHOP, Dr. Ashley Cook, who's at the FDA, and Dr. Peiyao Zhao, who's at the Allen Brain, who formerly worked for Dave Gilbert, and she's on the job market right at this moment and just an absolutely fabulous computational biologist. Another thing that we've done just in the very recent future is to think about disease as so-called nature's perturbation, and we wondered if we could use disease to get at, if we broke the 3D genome, what would happen to gene expression in a pathologic situation. And for that, we chose what we thought at the time was a classic monogenic disorder of Fragile X syndrome, which is known in the textbooks to be driven by a single mutation, which is a short tandem repeat track in the FMR1 gene in the 5' UTR. For reasons that are still poorly understood, this repeat sequence grows out of control. It reaches a length of 200 CGGs, and then it becomes full mutation. And at that time, there is so-called epigenetic silencing of the FMR1 gene and quite severe disease. So what the team had found is that indeed in Fragile X syndrome, there isn't just a local silencing of only FMR1, but in fact, in iPS-derived NPCs and neurons that have these Fragile X mutations, there is widespread megabase scale domains of heterochromatin placed across multiple autosomes. All of these locations come together in trans to physically contact each other, and the genes under these locations are largely synaptic genes that are silenced and have been linked both to Fragile X as well as to autism. Also, when we use these so-called locations to look at the base of those particular genomic coordinates, what we found is genome instability that we didn't expect in a monogenic disorder. And so we're calling them breaches or beacons of repeat expansion anchored by contacting heterochromatin. Again, these are inter-chromosomal interactions among large-scale heterochromatin domains. And what we thought was very exciting that happened over the last few months is that an independent group led by Marco Gallo in a rare form of pediatric brain cancer found a very similar pattern. They call them tulips, and it's the same idea that there are large-scale heterochromatin domains that silence genes that wouldn't be expected in another brain disorder. So we hope this begins to represent a pattern that could potentially mean we could have new ideas for druggable targets in thus far intractable neurological disorders. This work was led by a team of wonderful people. Thomas Malachowski and Dr. Ken Chandrados are still in the lab. Dr. Ravi Boya, who's in his last year and going on the job market at the moment, and Dr. Linda Zhao, who has completed her MD and her Ph.D. and is now in the late stages of dermatology residency at Penn. So during the pandemic, I think we all went through some form of existential crisis, and for me, I started thinking a lot about just the types of questions that the lab wanted to answer going forward once we were all able to be back together again. And I was deeply impacted by this book, an autobiography of sorts written by Eric Kendall, in which he describes sort of the broad history of neuroscience and teaches people new to the field, such as myself, the sequence of events or the historical events of many of the core discoveries. And it had us thinking about this concept of memory, and certainly the focus here was a lot on synaptic plasticity and neurophysiology. But at the time, the lab discussed it, and we felt that there would be very important ways in which chromatin could play a role in long-term storage of memory in neural circuits. And the reason is that memories, as we all know, are stored on timescales of decades. However, biomolecules, RNA, proteins, etc., have half-lives on the manner of hours to up to a few days. And so there has to be a connecting point that maintains things over the long term. I want to come back to this image that I showed you in the beginning. And one thing I wanted to share is that oftentimes the classic studies of neurophysiology and synaptic plasticity might overlap the contribution of chromatin to the storage of memory. However, we think that's because the timescales are so disparate. And yet, if we can come up with tools to connect the timescales, we can begin to study how the nucleus and the synapse begin to interplay to keep things going over the long term. This is not coming out of the blue. In fact, there's important critical data throughout the field that form the sort of foundation for IDEA. First and foremost, here is a classic EM study published in which looking at a single nucleus from a mammalian neuron cultured in vitro. Upon pharmacologically induced depolarization, we see marked restructuring of chromatin in that particular nucleus on what I would call fast time scales. And then upon removal of the stimulus, we certainly see recovery, as one might expect. But there are pockets that remain that, of course, we don't know from this image what that genetic sequence is, but we are very excited to figure this out. The other foundational data is that there's lesser-known studies that have looked multiple years ago now, almost 10 years ago, in CDCF knockout mice, but in adult animals. And, of course, we know that these would have defects in genome folding, genome-wide, as many studies at this point have shown. But these examples also demonstrated that there was severe learning defects, both in ability of animals to learn the Morris water maze, as well as in contextual fear conditioning. At the neurophysiological level, there were quite important defects in long-term potentiation. And then also at the cellular level, it has been reported that CTCF knockdown leads to a decrease in dendritic complexity as well as spine density and spine size. And so the defects are across scales, and we think there's a foundational basis for this. The other important point is just that in the classic literature, and even as summarized by Kendall, not only does any form of action potential lead to changes at the synapse, but there are also very rapid changes in the nucleus. Upon a stimulation event at the presynapse, of course, there's a release of neurotransmitters such as dopamine or perhaps glutamate. This leads to molecular and a sort of structural strengthening of specific synapses or spines, but it also causes the influx of calcium, which can kick off signaling cascades. These go rapidly to the nucleus to activate so-called immediate early genes such as FOS and ARC. And then also secondary response genes follow shortly thereafter. And so one leading very exciting theory in the field is that of Richard Morris and many other colleagues in parallel and after him, and that is of synaptic tagging and capture. The idea is that during the learning, there is the ever-elusive biochemical mark that stays at the synapse. This marks the spines that were originally potentiated. And then over the long term, cell-wide transcriptional programs, there are certain RNAs that go back to the spines. And those lead to the biochemical mechanisms that propagate those tags over the long term. And so we're excited to work on this problem and try to figure out some of these details among other colleagues that are already invested in working on this problem. And we've boiled it down to three things that we first have to prove to ourselves. The first is that we have to find something in the chromatin that's persistent. It has to be activity dependent in vivo, and it has to remain for a very long time, upwards of months or maybe even years. The second is that whatever that persistent thing is, it has to give us a flashlight or a beacon onto what RNAs might be changing in an activity dependent manner. And then those are our candidates to say, can we find the ones that make their way back out to the synapse? From there, of course, we would need to collaborate with protein experts to figure out what is actually going on for the propagation of the tag. But we think we have to find those two things. And then, of course, both of those things have to be disrupted to some degree in a disorder in which there is loss or gain of memory. So the lab now going forward is divided among 4 areas. First and foremost, we're studying how chromatin is changed in paradigms of synaptic plasticity in neural circuits, both in vivo as well as neural analogs of learning in vitro. We're actively building technologies, but now instead of in the genome, we're building single synapse sequencing and imaging technologies to create, over many years, synapse resolution, activity-dependent neural connectome maps, another connectivity problem. We also then want to study nucleus to synapse, so using our technologies to understand which RNAs head back out from the nucleus to the spines, and then once we identify their composition, how they might be acting, and are they functional for behavior. And finally, is the synapse and genome structure function miswired in specific neurological disorders? And so today I'm going to touch on 2 completely unpublished pieces of work. The first is just very briefly highlighting some of the things we've already published in the area of neural stimulation, and then show you new single-cell work from Zhu and Chandra Dose from my group. And then I'll also summarize a news story in a disease of loss of memory, Alzheimer's, led by Chandrasekhar Samandi, recently put on bioRxiv. So the original work in my lab in neural stimulation was led by John Began, and again, the idea is that upon calcium influx, when action potentials are fired, there is the activation of immediate early genes and also secondary response genes. And for chromatin structure function people, this is a very exciting paradigm because things happen so fast. Synaptic plasticity on timescales of milliseconds, immediate early genes on minutes to hours, and then secondary response genes also from on hours up until a day. And so this helps to get faster PhDs as well because the students aren't waiting for a year to process the samples. So I had no problem recruiting into this particular project, and this is just one example of data that John created. Here's the FOS gene on the x-axis and activity-induced enhancers as classically functionally reproduced by many neuroscientists in the field. And then these neurons are activated, and you can see within 20 minutes there is a long-range looping interaction that forms between this enhancer and a promoter. It stays for up to 60 minutes, and then it slowly begins to dissipate as the gene is turned off. And I wanted to show this time course just because in the early days we really had no idea if loops mattered for gene expression. So we measured the mRNA levels, and we saw they peaked around 60 minutes for CFOS, just as shown in the literature. And then we did the same time course for loops and weighed it up against the gene expression. And we're quite excited to see that loops actually form quicker than max mRNA levels. We saw peak loop interaction strength as early as 20 minutes, and we never went back to look even earlier. So we know that they form fast and they're activity dependent. I'm reducing perhaps 4 years of work down into a single slide, but I'll just say that we also saw structural patterns that might predict timing of gene expression. For example, FOS forms only singular loops, and it is activated super fast, and then it's also turned off just as fast. By contrast, BDNF, which is a secondary response gene, it forms a complex network of multiple looping interactions and quite long-range enhancers, and we think that might contribute to its further delayed gene expression. And so more loops, longer loops, and more enhancers, at least in a neural activation paradigm, often leads to delayed dynamic loop, gene expression. Another leading question in the field is, do loops matter for gene expression? And this was really highlighted by critical work that was kicked off by Erez Aden and his colleague Rao, as well as many others, at this point going up to 8 years ago. In this study, there was a degron that was used to knock down cohesin over very short timescales in a dividing cancer cell line. So cohesin was taken away, and no surprises here in this study in Erez's group, all of the loops were gone. What shocked the field and led to quite a bit of dismay was that in looking at the gene expression, and this was nascent transcripts through GROSeq, there was essentially no change whether these cancer cell line had loops or not. So loops don't matter for gene expression maintenance, at least in this system. One idea that we had is that we could still look, but we had to think about the establishment of new gene expression patterns. And so we teamed up with in vivo expert Matthias Merkenschlager and did this work across the Atlantic Ocean through the pandemic to knock down cohesin in mice and then to activate these neurons and to assess, do they have the ability to activate the immediate early genes and secondary response genes. And I'll just very briefly summarize, this is already published here, but the surprise was, first of all, that immediate early genes turned on just fine, even when loops were gone. However, a large range of synaptic plasticity genes, as well as secondary response genes, had no ability to turn on once we asked the neurons to do something new. So it's perhaps loops aren't that much needed if you just need to maintain what you already have been. But if you need to go somewhere new as a cell, then you will need new looping interactions to get to where you want to go next. So where are we going now? We're very excited about this. We're working as a lab with Erica Korb at Penn and Jason Shepard at University of Utah, two amazing neurobiologists or neuroscientists that also have amazing behavior models. And this work is led by Puebo Zhu and Ken Chandrados, two postdocs in the lab. And so we want to go in vivo and use contextual fear conditioning. And the idea is that we expose the mouse both to a neutral context as well as an aversive stimulus, in this case a Frochok. And now to begin to look for chromatin persistence, we have time points that span multiple days leading up to almost a month. We also then have a recall in which we expose the mouse about a month in to only the neutral stimulus, again, which leads to the same type of severe freezing. So this is a one-shot behavioral deal. And then we have isolation of neurons in various parts of the brain that are relevant for either the encoding, consolidation, or recall of fear memory. How do we get out all of these complicated patterns and get the neurons at the exact right period of time? And for that, we use an exciting and elegant mouse model that was developed by Lincoln Lowe at Stanford called the TRAP2 mouse. What this mouse does in brief is that it uses an activity-driven cray to essentially tag or genetically mark in a permanent manner the neurons that were fired only during the exact window of the behavior during the 4-OHT injection. And so then, subsequently, we can wait long periods of time and sort out those nuclei almost a month later because they were active only, to our knowledge, back at the original encoding. And the other critical piece here is we used a new multimodal single nucleus technology built by Joe Ecker as well as being run called single nucleus methyl 3C-seq. The power of this tool is that it assays both DNA methylation and the 3D genome. And so what we can do is now cluster the cells by their DNA methylation profiles genome-wide. And we can grab all of the neurons that look exactly like they have the same exact DNA methylation profile, and therefore they are likely a matched excitatory or inhibitory subtype. That gives us the ability to collapse back the single-cell high C-maps to create a matrix that's less sparse. And therefore, computationally, it becomes tractable, again, to call loops in single neurons, but now a proxy because it's a pseudo-bulk measurement. So this is just data. This is hot off the press just in the last 2 weeks, coming out from 2 to 3 years of effort by both Puebo and Ken. This is around the NMDA receptor gene. And what I can say is that here is a location where there is no loop form in these neurons in the control. Then we see a loop is formed in short time scales in the trapped neurons. It remains at 3 days, and then it also remains through day 28. And so this is just one example, just in the last few days, where we think as a lab that we are going to be able to have at least the sensitivity to search for and potentially find persistent looping interactions. So our idea here, or the hypothesis that we're chasing or trying to disprove, is the idea that during memory encoding there are the formation of activity-dependent loops that stay stuck and they're persistent. They remain over long periods of time, even if the gene expression comes back down. And then when that neuron is called upon again, perhaps through a retrieval or a recall, that in some way poises these locations to reactivate the genes, but in a way in which there is a faster response, and we'll have to figure out the functionality of how that persistence enables this re-expression during recall. So just to summarize this part before going to the last section, we find that in neurons, many loops connect de novo to activity-dependent genes upon neural stimulation. These loops are fast. Immediate early genes connect to enhancers in short-range loops. They occur as fast as 20 minutes, and they peak prior in strength to maximal mRNA levels. And we also see they're cohesin-independent. We also find the secondary response genes engage in a complex network of long-distance loops that connect on later time scales, and they're certainly cohesin-dependent in vivo. Cohesin-mediated long-range loops are necessary for the establishment of neural maturation and synaptic plasticity gene expression in vivo, and activity-dependent secondary response gene expression in vitro. So for the question of do loops matter, at least in post-mitotic neural activation, they certainly do, but for the establishment of new gene expression programs. And then we also find through new preliminary data that I've just shown today that persistent loops in engrams may exist, and we're continuing to pursue that through the final year of the 4D nucleome. So now I wanted to switch gears and go over to the red part of our lab's focus areas and show you unpublished data from Harshini and Zoltan. But now in thinking about, well, what about in a disease where there is loss of memory? And for that, we turn to Alzheimer's disease, which there are many experts here at the NIH. I had the great fortune of discussing this with someone earlier this morning. It's the most common form of dementia, of course, and plays out through a complex sort of temporal events of neurodegeneration. As many of you know, the large majority of cases are sporadic, 95% or more, and they're late onset. But there is a subset, about 1 to 5%, that is early onset, driven by autosomal dominant rare mutations in the APP or PSEN genes. The commonality between these two is that they often have a similarity in biochemical hallmarks of either aggregates of amyloid or hyperphosphorylation of tau. So something that's been less appreciated is that the multiple cell types in AD also have severe transcriptional defects, and this has lately come into fashion because of beautiful single-cell studies done in postmortem brain tissue. But one thing that's often been a question, well, what type of mechanisms participate in AD in informing this widespread gene expression dysregulation? Why has there been less insight in this area where there's many critical limitations? The first is that many studies, and rightfully so, are focused on AD patient-derived postmortem brain tissue, but the neurodegenerated neurons may already be dead or in late stage, so it's very difficult to get back to the early events. So molecular and cellular defects that occur decades before the clinical presentations will simply be missed by focusing on that late stage. Late stage, again, misses early drivers of the disease. And then at least for sporadic Alzheimer's, IPS neuron studies are difficult because the degeneration is late onset and more sporadic and variable across individuals. So it's just very technically challenging to capture the details in a reproducible way in neurons that often in a dish are representing a fetal stage. So our goal as a lab is that we wanted to see if there was chromatin dysregulation linked to gene expression changes during the early onset of AD, and we wanted to get time points that were before there was obvious presence of either amyloid plaques or tau tangles. So this work was led by Harshini Chandrasekhar, who has now defended her Ph.D. and on the job market currently, amazing computational biologist, coupled with Zoltan Samandi, amazing wet lab postdoc, and then supported by many other important members of the team. It's been a long-time collaboration with friend and colleague Dominic Piquet, who's at LMU Munich. So to do this work, we use Dominic is an expert in creating IPS lines of Alzheimer's, and we wanted to focus on familial Alzheimer's disease to get that early onset mechanism. And so Dominic has created these karyotypically stable lines in which they're isogenically matched for their background, and he and his lab have engineered in mutations, autosomal dominant mutations, but in both alleles, so homozygous in these IPS lines. From there, we used a 2D monolayer differentiation protocol that Dominic and his team have optimized over the span of 10 to 20 years to get a pure population of so-called excitatory neurons. And then in parallel, we also created brain organoids through the protocol of Sergei Pascha to look in both 2D and 3D models. And what was important with these lines and that we were excited to see is that when we take the organoids created from these IPS cells out to day 200 in vitro, we're able to see the biochemical hallmarks of early onset Alzheimer's, including aggregates of amyloid as well as hyperphosphorylated tau. But with those same IPS cells with that same genetic background in 2D monolayer, when we culture them up to day 65, we do not see the presence of those hallmarks. And so for us, this meant that we could get a pure population before we see the obvious biochemical hallmarks. And we wanted to look in the 2D situation to ask if there were severe chromatin defects already early before the amyloid beta changes. So first and foremost, we did gene expression and looked at RNA-seq, and we looked across the time course of differentiation of early and late NPCs as well as post-mitotic cortical neurons. And what was notable is that with the APP-SWI rare mutation or the PSEN1 mutation, we saw widespread gene expression dysregulation with a fourfold more decrease in gene expression than increase. This result was reproducible across different mutations, so the same genes that were down and up in APP-SWI in large part were also disrupted in PSEN1 mutation. And then also when we looked at day 100 in the organoids, we saw also a similar pattern. And I'm not sure if I showed it here, but then this completely changed to a different pattern at day 200. Initially, we wanted to look at chromatin accessibility, and there were a few surprises here that we're still trying to figure out. First and foremost, that all of the genes that showed dysregulated gene expression, when you looked at the chromatin accessibility at the promoters, 99% of the time, it did not change at all, even though the genes were dramatically changing in expression. However, when we looked at distal non-coding regions, those were the locations where the chromatin accessibility changed between genotypes, but in ways that were not really that predictable with the gene expression. And so for a lab that was focused initially on the 3D genome, this gave us insight that we needed the loops to figure out this problem. So here's an example of our Hi-C in the post-mitotic neurons, both in wild type as well as the APP-SWI genotype. And I'll just give you an example here to see the dots around this gene and then how they're dramatically disrupted in post-mitotic neurons. And we called these quantitatively with methods that we've developed within our larger community over the years. We found roughly 1,700 loops that are gained in post-mitotic neurons with this rare mutation, about 30,000 loops that are completely unchanged, and then a little bit over 3,300 loops that are lost due to in the genotype with the APP-SWI mutation. All right, so back to this question of do loops matter. I think this has been like the crowning question of the 4D nucleome, and I know that many of you are also asking this in your model system, and we wanted to use our data to answer this and get direct observations that I could show you here today. And so we subclassified our loops into those that are enhancer promoter and that they're lost in post-mitotic neurons due to the rare mutation. We also grabbed those loops that are gained due to the rare mutation. We found the loops that were unchanging. And then we also grouped up genes that were not forming any structure at all. And then we wanted to ask, so now what happens in gene expression in this non-dividing post-mitotic situation? So there are a few surprises, but this one you will not be surprised about. First and foremost, when you see a gene that loses only one loop in post-mitotic neurons, there is essentially no predictive power for what is happening with gene expression. You can have equal probability of being unchanged, increasing expression, or decreasing. And this is representative of the disparate experimental results that have also been demonstrated in our field. But what we did see is that when we begin to stratify genes by the number of enhancer promoter loops that they form, the more loops that you lose, the more it's predictive of the directionality and the effect size of the lost gene expression. This result was not just with lost loops, also with gained loops. The more loops that a gene gains, the more it's predictive of a gained or upregulated gene expression pattern. No surprises here. If the loops are unchanged, there's really no predictive power over changes in gene expression. And also genes that don't loop at all, there's also no predictive power. So as we like to say in the lab, more loops, more problems. Okay, what is at the base of all these loops that are changing? I'm going to spare you the many defeats we had as a lab and just say for aficionados that among the architectural proteins and the obvious candidates, we looked at the base of the loops and classified them in all sorts of ways. And the bottom line is that all of these loops that are lost and gained in post-mitotic neurons, they have CDCF, they have cohesin, it is unchanging. This puzzles us and continues to because there is something else that is leading to these loop dysregulations. And the one main signal that we've been able to find over the years is that at the base of these loops that are both lost and gained in this Alzheimer's mutation genetic background, they have very strong presence of RNA Pol2, not only at the gene body, but also strong at the enhancer. And then this is lost when the loop is lost in FAD. So luckily, because it's Pol2, there's a very tight peak, and we can look under the base to say what motifs are at that location. And we did this work for both the lost and the gained loops in these APP-SWE neurons. Usually with motif enrichment analysis, as many of you know, you can find any number of hits with this type of work. But in this case, we just kept getting one singular hit, a motif TAA-TTA. If the loops are lost under those Pol2 dynamic sites, it is present in an array of many sites adjacent to each other. If the loops are gained, it's present typically in only one copy. And when one looks, the result that you get is fairly disturbing. These are homeobox transcription factors that, of course, are highly redundant and therefore would be almost impossible to do a singular loss of gene experiment. But there are a few that stood out to us, DLX1, DLX2, DLX6, and LHX2, and we searched our gene expression results. And here's where I think we got a little bit lucky. We saw that, for example, across our differentiation time points, the gray is the wild-type neurons and the orange is those with APP-SWE. The LHX2 mRNA plummets in neurons and is completely turned off, but it's really high in wild-type. Similarly, but opposite, DLX5 and DLX6 are not present in wild-type neurons, but strongly express, many-fold change upregulated due to the mutation. And so for that reason, it gave us candidates that we can begin to pursue with gain-of-function studies. So what we're doing right now is overexpressing DLX1 with the HA tag so that we can chip it, and we're looking at the consequence in a time course on Hi-C and on gene expression, as well as the cell fate of the neurons. I also wanted to share that with great excitement, we saw recently a new preprint dropped by Evan-Jean Kwan in collaboration with Panaccio and Vizel, now in a completely different model system of the sonic hedgehog locus in vivo. And in this study, what they were looking at are distal enhancers and what might encode or assist in the looping interactions. And similar to our work, but in a quite different system where they can do these perturbations in vivo, what they found was the same motif, TAA-TTA, participating in the formation or disruption of looping interactions. And so we're doing the gain of function by overexpressing in an in vitro neuron model. They are doing the CRISPR mutation of the mutations in a limb-bud in vivo model, and we're trying to attack this question from 2 angles in collaboration. Okay, so I want to end here by bringing this forward as something that I can say John Satterlee and many of the program officers have really challenged us over the years, and rightfully so. And that is, at least in the larger 40 nucleome community, we have spent a lot of time and effort and blood, sweat, and tears building these computational and experimental technologies to map and understand how the genome folds. And at the end of the day, even if we think it matters for gene expression, what is the physiologic significance? And so we wondered if there could be something within our system that could help us understand. We looked first at our neurons that we know have massively disrupted loops as well as gene expression. And those who are microscopists might find this disturbing, that indeed we saw absolutely no apparent changes in cell density or neural morphology. Where the insight came is when we began to do more analysis at the single cell level. So we did single nucleus RNA-seq for the neurons. And what we found is that, no surprises here, in large part the wild-type neurons were excitatory in character, just as Dominic Piquet's protocol had already published. However, when we looked in the APP-SWE mutation, although not obvious by morphology, the neurons now had completely shifted their composition, their substate, to now be almost entirely inhibitory in character, but we wouldn't have detected it by eye. So this is very surprising to us, and we're doing subsequent follow-up studies now. But the emerging model from our work is as follows. In wild-type human neurons derived from iPS cells, we use CRISPR to add homozygous, rare autosomal dominant mutations that have been causally linked to early onset Alzheimer's. Upon the differentiation process, what we see is that there's many long-range looping interactions, and these can be disrupted due to the mutation. At the base of these disrupted loops are this motif at the enhancers TAA-TTA. Then we also see gained loops in the same model system. They also have TAA-TTA at the base, and we're pursuing the gain of function overexpression to assess causality. And what we're excited to say is that if you look at the base of these loops, it's oftentimes excitatory and inhibitory genes that are disrupted. And then what we also see is an imbalance. So in this wild-type state, there is mostly excitatory neurons, and then it shifts in parallel with this change in loops in gene expression to be more inhibitory in character. And we think this might be one important place where loops and the gene expression that they control might matter for the cellular state of neurons in human disease. So our data uncover a possible genome structure relationship between multi-loop miswiring and dysregulated excitatory and inhibitory gene expression. And one exciting thing is that it appears, at least in our model system, to occur prior to the deposition of biochemical hallmarks of amyloid aggregates and hyperphosphorylated tau. So I have about 3 minutes, and I just want to close here with maybe 3 slides of where we want to go in the future. This was a beautiful editorial that summarized the work of some of my colleagues, in which they used simulations to just sort of capture the imagination of how difficult the 3D genome folding really has been over the last 10 years. I am showing you here today some things that have worked, but I have to say that in those early days, we did not know if this was going to be a tractable problem. And oh man, it was hard. But it was fun, and so now I want to do it again. And the idea that has really called out to us, I think, is just another connectivity problem of the neural connectome. And we've started the process of thinking about how we want to use the principles, both computational and experimental, that we have learned through proximity ligation and Hi-C, but begin to think about ways that we could do molecular maps, but now of the synaptic connectome. And so that's what we're working on now, thinking about building SynGram-seq to create synapse resolution activity dependent connectome maps. Of course, this is going to be quite hard, but we're excited to work with some new young people with new ideas to begin to tackle this problem. The other one, as I've alluded to multiple times, is that even if there is persistent chromatin, whatever it is helping with has to get back out to the synapse. At least we think so at this time. And so developing and applying single synapse technologies to find which RNAs they are and how they change in a dynamic manner, and then proving those RNAs matter in some way for behavior. And then finally, although I've shown you an example of Alzheimer's today where I focus almost entirely on chromatin, we hope to sort of revisit those studies, but to understand how those loops might matter for how the synaptic plasticity might be dysregulated in disorders like Alzheimer's or Fragile X. This is my last slide, just to say that our interest is in focusing on a few model systems and really thinking across scales from the nucleus out to the synapse. And our hope is that over time, we can, perhaps in the next 10, 20 years, find some insights that can give genuine help for many that continue to suffer in the myriad of problems the world faces in this post-pandemic world. With that, I'd like to close and just really, first and foremost, highlight the work of my group, both past and present. It has been a privilege to work with them every day, and they keep me young and they keep me excited. And just thank you to all of you online or even in the future who listen to this for investing in our lab and working with me. And then also very important, as John has alluded to, just thank you very much to the NIH and colleagues at the NIH. And I think just those small, consistent steps of support, maybe in one single instance might not affect a person as much, but over the years, as that is sustained, I think it just has an outsized impact on people, and just appreciate your support over the years, and then how it has impacted all those in my group. So thank you again for having me, and I'll be happy to answer questions.
Thank you. So, of course, anyone here should be using the mics. I can kick it off with we have a robust online audience. Here's a question. I'm going to rephrase it. Can any or some of these experiments be done with the surgically ablated human samples?
Yes, we do believe that these experiments can be done with the surgically ablated human samples. Perhaps not some of the genome engineering, but many of the single-cell technologies, we do believe that's possible. I do think to have real insight, we're going to have to layer on the circuit information, so tracing of the circuits and getting out not only the trapped neurons that are active during a behavior, but also only those that are connected. It's getting to the physiologically relevant neurons that, for us, has been the most difficult piece and is going to be so critical. And then it will change with every behavior, with every experimental, every experience. You've got to get it just right. And luckily, there's so much beautiful work out there to say, okay, what circuit should we get at? And now we've got to trace it and get it.
Hi, very interesting talk. I'm very intrigued by your the final presentation about iPSC-derived neurons with the APP or presenting single mutations. And I'm wondering whether you have done any like heterozygous mutant cell line that showed a similar kind of change of the neuron, like a more inhibitory neuron now.
Yes.
The first question. The second question is, I think people have already made some kind of homozygous knock-in mice for these mutations, but I don't really remember that dramatic change of, the neuron presentation, the fate, the excitatory inhibitory. That effect is huge. So just say, besides all the in vitro work, have you verified the finding with the in vivo model?
Yeah, thank you very much for your question. So there's two parts, and I'll start with the first one, summarize for our live online audience. So the question is, we use homozygous mutation of a rare autosomal dominant mutation. So what that means is we did that on purpose, and the idea is that we wanted in our first pass, because these studies are so long and so resource intensive, we wanted to get the highest signal to noise that we possibly could get. And then if we didn't see anything, we would move on to a different study. And so when we did that, we saw severe changes. And as you saw, the excitatory to inhibitory shift was dramatic and severe. And I think what the speaker, the questioner, even though they didn't raise this, was probably alluding to is that in early onset Alzheimer's, you do not see this dramatic change to entirely brains of entirely inhibitory neurons. And we think the reason why we see it to that extent in our model system is we're putting in 2 copies of this rare autosomal dominant mutation, which to my knowledge is not present in human populations. So then they ask, have you done these studies with a het to simulate what it really happens in nature? And we haven't done it yet, but we are working with Dominic to validate those lines and ensure their karyotype instability. And what we anticipate is that there will be far less loops that are a problem, far less gene expression dysregulation, and only a slight shift, which means we have to use only single-cell technologies to look. The second part of that question was really a different aspect, which is thinking about mouse studies that have, if I understood correctly, was a humanized mouse that knocked in perhaps a human APP gene or PSEN gene with the mutation, and saying, could we look back at that literature and look into the characterization of the excitatory versus inhibitory population? That's an excellent suggestion, and we haven't done it yet, but it's something we can do.
I have a question about your looping work. So you showed that when you only lose one loop, there's no changes in gene expression, and then the more loops that you lose, you see a decrease in gene expression. And I was thinking back to the CTCF deletion and how we didn't see any changes there. Are most genes regulated by like 1 or 2 loops? Is that how that works, or am I misunderstanding? How would I reconcile these two?
Right. So I think you're referring to our work collaboration with Matthias Merkenschlager, where we knocked out cohesin, actually, and then looked at the disruption of loops.
Yes, and you saw no looping, and the gene expression was...
That's right. So for people online, there's a question to clarify the in vivo work with Matthias Merkenschlager versus the multi-loop model that we're seeing in Alzheimer's. So just to clarify with that in vivo work, which is in eLife, if you want to look up the paper, what we saw is that when you knock out cohesin, there actually is severe gene expression dysregulation during neural maturation in vivo. And then what we did is we took the neurons out, and in vitro we used pharmacological agents to tell them to fire action potentials. And what we saw is that loops didn't really matter for immediate early genes, which are the ones that fire fast, like within 5 minutes. But then when we go to secondary response genes, which are fired on the timescale of an hour or later, or synaptic plasticity genes that are activated much later, that's when loops mattered quite a bit. And so it was more about the timescales, and loops certainly mattered. Now, in that case, we did not look at the multi-looping idea. It was published before we had found it in this later study, and so I can't comment on at this time if multi-looping is a phenomenon that matters for activity-dependent expression.
Do you see any plaque buildup eventually in the 2D neuron cultures?
Yeah, so the next question is about the FAD, and the question is if we wait long enough in 2D, do we eventually see plaques or hyperphosphorylated tau, although you didn't ask that part, in the 2D monolayer neurons. You know, probably not surprisingly, I never even tried to convince the students to keep those neurons going for another 100 days. And I think even if I tried, the answer would have been no. But I didn't have to try because there's beautiful historical work already out there that has been very difficult with IPS models of Alzheimer's in 2D flat culture to have an aggregation of something that resembles an aggregate, an amyloid aggregate. So we already thought that that was going to be intractable, and we're very fortunate that in trying the organoids within 200 days, we did see it. There was concern in the lab at 100 days when we harvested those organoids. There was nothing, and we were getting nervous at that point.
Thank you.
Hi, that was a very interesting talk. I was trying to connect the dots between the different parts of your talk. And there was one part of the talk where you were emphasizing the importance of the forming of these different loops and connections between different parts. And I was not sure whether that was in connection with just random activity or with learning, but it was it learning or is just random activity?
Right, so sure. The first question is, in the case of the activity-dependent looping, Is it related to, if it's in vitro, is it spontaneous firing? Is it evoked action potentials? And then when we showed the in vivo data, was that from behavioral or experience-induced learning, or was it some sort of optogenetic stimulation? So I can clarify, for our in vitro data, that is pharmacologically induced action potential firing, not spontaneous. And then our control is TTX. So we ensured, using that pharmacological agent, that we attenuated spontaneous firing as much as possible. For the in vivo situation, we used contextual fear conditioning, and we used that behavior and for OHT injection in the TRAP2 animal to only get neurons that were firing action potentials during the window of the behavior.
Yeah, so the question for me was, so that was cool and exciting, and then you talked about Alzheimer's and making organoids. So, and then a lot of the genes, mostly I recognize them as developmental genes, though they may have other roles.
Yes.
And so I was trying to figure out the connection between loops that you had established in the context of actual neuronal activity and perhaps learning or some task, and this thing that's happening in an organoid, which may have nothing to do with that.
Yes, thank you. So this is an important question, and for those online, it's related to how do we connect some of the work that is happening in circuits with now the in vitro human model of Alzheimer's disease, in which we are clearly and quite surprisingly seeing changes in developmental genes and developmental looping interactions. And what I have to say is that our plan in the future is to now, in that Alzheimer's model, begin to look at action potential firing. We certainly didn't do it for this study that I showed today, but that's the direction that we're going, and you could probably guess that's why we're pushing toward organoids. We're also looking into the 5X FAD mouse to see if we could do something in vivo as breeding it with the TRAP2 animal. So at this point, one thing that I will say, and certainly on the record, is that there was no one more than me more shocked by the widespread nature of the loop dysregulation and the fact that it was a lot of developmental and early synaptic plasticity genes. And it is likely partially part of the model system because we have a homozygous mutation. But I think there's real insight there because you are starting to see in conferences across many neurological disorders that although the cognitive defects are manifest very later in life, there's starting to be this movement of how soon do we have to look in development and early adulthood to see the initial unraveling and the initial onset of disease. And I do think it's possible that over the years we're going to start to see that defects at the molecular level happen a lot sooner than some of the severe clinical presentations. So we're going to keep pushing it, and as somebody else mentioned, maybe bringing in the HET will help us understand effect size instead of high penetrance of the effect.
I should have stayed with the mic, but so what you found is very interesting, and it predicts or it suggests that people who have this kind of Alzheimer's are likely to be associated with developmental problems that could be identified way earlier because there's already a change in the balance of the number of inhibitory neurons. I can't imagine that would be without consequence.
Yes, so there's an important idea raised by the questioner about if individuals with early onset Alzheimer's, some of them who might have this molecular defect that I showed today, might also manifest in the clinic as having presentations that are more typically equated with neurodevelopmental disorders. You know, I'm not a clinician, so I don't know if that idea is heresy to other experts in the field or not, but it's something that we would be excited to keep our eye on. And I know even here at the NIH, we're looking broadly at many different quantification of features, both molecular as well as clinical presentations, and I think it would be very exciting if that ended up being the case.
Maybe one last question, and we are past the hour, but please.
Yes, my name is Mike Mage. I'm 90 years old, and for many years my lab chief was Maxine Singer, who unfortunately died this summer. And I would like to challenge your time frame which you mentioned of a decade, when I look on TV at the news and I look at the pharma companies which are using monoclonal antibodies to identify sequences which are relevant to Alzheimer's and which are using artificial intelligence enhanced sequencing to find low molecular weight drugs which influence Alzheimer's, and that I would suggest that if you look at those carefully and see what they are doing, you will see that your time frame should really be on the order of months rather than on decades.
Thank you.
Thank you so much. Thank you, Jen. I'm here to tell you all that I'm told there's a reception after this, and there will be cookies and possibly Jen's favorite chemical, caffeine, possibly. So we would like you to join us to talk to Jen during the reception, and if you could all join me in a round of applause for an awesome talk.